I'm going to talk to you about a coronavirus this morning, and coronaviruses have been in the news a lot lately. So I'm going to go through my presentation, and then at the end I'll have a couple of slides just to update you on what's been going on in the news. But this is the outline that I'm going to follow this morning, and just really briefly go over what this disease is that we study in chickens, the characteristics of the coronavirus, some of the challenges that we have for controlling this virus, and then we'll go through some of the new methods that we've been researching in my laboratory for its control. So IBE is caused by an avian coronavirus. Coronaviruses are highly contagious. Morbidity is always 100%. Mortality can vary quite a bit. And this is due to secondary bacterial infections that can come in after the virus causes the damage in the upper respiratory tract. It is worldwide in distribution. It is a chicken disease, so everywhere we raise chickens in the world we'll find this particular virus. And there's multiple different antigenic types of this virus, which makes it really difficult to control. The clinical signs, it's really a mild upper respiratory disease. It causes coughing, nasal discharge, conjunctivitis. Rales is this gargling noise that we hear when birds breathe because of the mucus in the trachea. It can also cause nephropathogenicity or nephropathogenic strains. And this is where these viruses can infect the kidneys of the bird, and we'll see flushing of the birds, we'll see uriates into kidneys. And it can also cause reproductive losses. So in laying birds, when laying birds become infected with this virus, we'll see a drop in production, and we'll see these misshapen and wrinkled eggs resulting from this. So it is an RNA virus. The genome of it is a single-stranded, positive sense RNA genome. It has 4 structural proteins: the envelope, membrane, nucleocapsid, and spike. And it's the spike protein on the surface of the virus that's the one that's the most studied. It's the most important protein on the virus. This is the protein that allows it to attach and enter the cell in the bird. And it also is the protein that induces neutralizing antibodies. So when those antibodies bind to the virus, they'll protect the bird. Now, I mentioned that there's all these different antigenic types of this virus, which is a challenge for its control. And the spike is what is involved in those different antigenic types. So when we develop a vaccine, we want to induce neutralizing antibodies to the spike. But because all the spikes are different, the antibodies induced by one type of the virus may not bind very strongly to another type of the virus, and those birds may still be susceptible to it. A lot of work's been done on spike. There is a minimal binding domain, which is the white part of this particular model. And then those, the red amino acids that you see highlighted there are critical for its binding to the host cell. Okay? But changing just 1 or 2 of those amino acids will prevent it from binding, but it also changes the structure of the protein itself. So when we induce those neutralizing antibodies, it won't bind to that particular spike anymore. So developing vaccines is really a challenge. We talk about different types of this virus, and we talk about 3 different ways to type it. The traditional method is by serotyping. This is where we develop antibodies against the one virus, and we ask the question, will it neutralize another virus? If it will not, then that is a different serotype. Now, with the advent of molecular testing and sequencing, we've developed these genetic types classification for this particular virus. And this is the way most folks are doing it now, because it's really fast and it's easy to do. And this is basically defined by the sequence of, you guessed it, the spike protein that's on the surface of the virus. And then the last way we type these viruses is what we call protector type. And this is a term that was coined many, many years ago. And this basically is when we ask the bird whether it's protected or not. So we'll vaccinate the bird with one type of the virus, and then we'll challenge with a different type. And if the bird is protected, then they are considered the same protector type. If they're not protected, then they're different ones. Serotype and genetic type correlates pretty well with protection. But because the bird responds in many different ways to vaccination, we have innate immunity, we have cell-mediated immunity, we have antibodies that are being produced. The prediction of a protector type is much more difficult. We really have to do those studies in the laboratory to determine whether what bird is going to be protected with this vaccine or with another vaccine against the circulating field viruses. Now, one of the challenges with coronaviruses is they change very rapidly because they are RNA viruses. They can undergo mutations when they replicate, and this virus in particular can also undergo recombination. When we have mutations occurring, that's called genetic drift, and that's where the virus changes slowly over time as we accumulate these mutations in the genome. When we see a genetic shift or a very quick change in the virus is when we see recombination events occurring between two different parent viruses, which will give us a different progeny back out. So the slide is just to show you kind of an idea about how many different viruses we have circulating right now around the world. And some particular problem viruses that we have are Arkansas DMV 1639 and Georgia 08 here in the United States. Worldwide, well, mostly in Europe, N162, but worldwide Q1 and the QX virus are quite problematic. And we have very few vaccines actually available to us for all of these different types. So how do we control this virus then with all these different bronchitis virus types that basically don't cross-protect? So local immunity is really important. Okay? This virus enters through the upper respiratory tract, so we need to protect that part of the bird. Okay? So we're looking to develop antibodies and cell-mediated immunity in the upper respiratory tract. Okay? So we vaccinate to prevent the disease, and the ideal situation is to block the infection. And with a homologous vaccine, we can get what's called sterile immunity, where we actually block the infection in the bird. And this is ideal because we then don't allow the virus to replicate, because every time it replicates, it can induce these mutations and we can get different mutant variants coming back out again. The other thing we try to do is if we don't have the perfect situation to block infection with a vaccine, we want to try to reduce the replication and prevent this virus from transmitting. So there is an infectious dose that we measure with these different viruses, and if we can get the virus load below that infectious dose, we can allow the virus to sort of burn out. Okay. So this sort of is just a life cycle, if you will, of this disease, and the first thing that happens is we get infection. Upper respiratory tract infection. That virus then replicates. While it's replicating, we see substitutions, insertions, deletions, recombination can occur, and we've documented that this actually can occur in one replication cycle. This virus changes that quickly. It develops genetic diversity. That's what it does to survive within its host. And then if enough genetic diversity develops as it passes from one bird to the next, to the next, to the next, we'll get the emergence of a new strain. If we get the emergence of a new strain, now the bird's not vaccinated against that particular strain, we can get the disease all over again. Okay? So with vaccination, what we're trying to do is we're trying to block the infection. That's the ideal situation. But if we can't do that, we want to block or we want to reduce replication to the point where we can stop the transmission of the virus. And if we can do that, we'll allow it to burn out and we can control the disease. So vaccination strategies for this particular virus, when a homologous vaccine is not available, what we try to do is we try to develop a broader immune response. And the way we do that is we vaccinate with more than one type of the virus. Okay? When we give two different types of the virus, or sometimes even 3 or 4 different types of the virus during the bird's lifetime, we can develop a broader immunity and we can help to protect against some of those variant viruses where we don't have a specific vaccine. The other thing that we can do is we can use 2 separate vaccinations. So we can use a prime followed by a boost. Okay? And we usually use, say, 2 vaccines in the prime and then 2 different vaccines in the boost, and that will also broaden our protection. So live vaccines induce this local immunity. That's the most important thing we're trying to do because we want to block the infection. Okay? Killed vaccines are not as effective because they don't induce this local immunity. Killed vaccines are used in long-lived birds to develop maternal antibodies that can then be passed on to the progeny. But the thing with killed vaccines that we have to remember is that they're really not effective unless we have a live prime given prior to that killed vaccine. So I asked the question, what can be done when a vaccine combination or vaccine types don't protect against the variant virus? Well, we need to create a new attenuated vaccine, homologous to the variant virus. Okay? So the way we traditionally have done this is by taking that field virus and passing it in embryonated eggs. And it needs to be passed anywhere from 80 to even over 100 times. Okay? Now, if you're really good and you don't contaminate anything and you don't screw up, you can pass the virus twice in 1 week. 52 weeks in a year, you can do the math. It takes about a year to attenuate one of these viruses. Okay? Then the testing and licensing that we need to do to get it on the market can take anywhere from 3 to 4 years beyond that. So we're talking about 5 years for a vaccine to come to market from the time we start with the attenuation process. Now, the challenge here is that these variant viruses, they may only circulate in the field for 2 or 3 years. So we've embarked on a mission to create a vaccine against one of these variants. It's going to take us 5 years to get it to the marketplace. That virus may be long gone by the time it's licensed and available for use. Okay. Variant viruses in the field also seem to change and emerge about every 5 to 8 years. So every 5 to 8 years, we see a new virus that emerges in commercial poultry that we have not seen before. So basically the overall point of this talk is, what improved methods do we have to be able to control this virus? Well, homology in the spike protein is real important for protection. We need a homologous vaccine to the field viruses that are circulating there. So most of the work that we've been doing and that others have been doing in this area are to try to improve the methods to focus on more quickly attenuating the virus. So we can take that year's period of time and shrink it down to a much quicker period. Okay. I can tell you what doesn't work. Okay. Vaccine vectors have been used for things like Newcastle disease and infectious bursal disease virus. They do not work with infectious bronchitis virus. And this is because that spike protein needs to have the proper folding, that three-dimensional structure, to be able to induce the antibodies that will neutralize the virus. So we take that gene out of the virus and put it into a vector like pox or HVT. It just doesn't reproduce that protein correctly. DNA vaccines, they give some partial protection, but you need to inject a lot of DNA and you need to do it multiple times. Subunit vaccines, they just don't work at all. We don't have that protein in its proper folding to be able to induce the antibodies that we need for neutralization. So what has been done fairly recently, I say within the past 5 or 10 years, is making autogenous live vaccines to this particular virus. Now, autogenous vaccines have typically always been killed vaccines because killed vaccines are safe. We can take a virus out of the field, we can grow it up, we can kill it, and then we can give it back to those birds on that same farm. More recently, autogenous live vaccines have been developed for infectious bronchitis virus. Now, what this allows us to do is short-circuit some of the licensing that needs, or the testing that needs to be done for licensing. So we still have to attenuate it, but then because it's an autogenous vaccine, we can get it quicker into those sick birds. So autogenous live vaccines are produced under a USDA license from Title IX. It's shown there. They fill a niche that's created when a vaccine company doesn't really want to invest all the time and money to produce a vaccine the traditional way. Okay? Relatively new because traditionally, like I said, autogenous vaccines are usually killed vaccines. They are used for bronchitis because we want that local immunity. We need a live vaccine to create that, to be able to replicate in the upper respiratory tract and induce that immune response. And again, killed vaccines have a very limited ability to do that unless we have a live vaccine first. Okay. And these attenuated vaccines, they're attenuated in a traditional way. Safety testing is kind of minimal because we want to try to get it out into the market very, very quickly. So there are safety and efficacy concerns with some of these live autogenous live vaccines. One of the things that we have explored and have been done by some vaccine companies as well is what's called heat treatment attenuation. And this is a method to short circuit the attenuation process so we don't have to take a full year of passing in eggs over and over and over again. Basically, it's just a rapid method to attenuate the virus by exposing it to heat. So we need approximately 10 to 15 heat treatment passages, if you will, in embryonated eggs to attenuate the virus. Then we usually have to pass it a few more times to be able to get the titer up high enough to be able to actually test and make a master seed from that. But this really shortens the time that we need to be able to attenuate these viruses. One of the drawbacks here is not all viruses, not all the bronchitis virus types lend themselves to this heat treatment attenuation. So it's kind of it's a bit of a hit or miss, but it's a first step in creating an attenuated virus because it is so quick to do. Once we have the heat-treated attenuated virus, we can produce a master seed with it, and in a very short period of time, we can produce those attenuated viruses that can then be used as an autogenous live vaccine. Another way to short-circuit this attenuation process is something called an infectious clone. Okay? So an infectious clone is basically a copy of the viral genome that we maintain in the laboratory. Okay? And we can then modify that particular genome so that we can change the spike gene to match the spike that is on the virus that's circulating in the field. Okay? So the backbone that we start with is an attenuated virus. It is a vaccine virus. And then what we're doing is we're just putting in the spike that we want. Okay? So this is just sort of a schematic of what an infectious clone sort of looks like. The upper part of this figure is the genome of the virus. It has a T7 promoter on the end of it, so that on the left-hand side or the 5' side, so that we can then recreate that RNA genome. It has a poly-A tail on the other end, which mimics what the virus looks like. And we've cut this thing into 4 different pieces, which overlap and have restriction enzyme sites, so we can stick it back together again. Okay. So the way this works then is we have a variant virus in the field. We sequence the spike gene of that particular variant virus. And then we don't even have to isolate the virus in eggs. Since we've got now the sequence of the spike, we can just synthesize that particular gene. Once it's been synthesized, we can then insert it into our infectious clone, which again is a backbone that is already attenuated. It's a vaccine virus. We then rescue that infectious clone with that new spike in it that matches the virus that's circulating in the field. And we can do all this in about 2 months' time. So really, really quick. We know we've got a safe vaccine. We can test it really quickly to make sure that it's efficacious and it could be used. Now, the stumbling point here is USDA licensing. They've never licensed a product like this before. So getting something like this through licensing the first time may be a bit of a difficult situation. We're hoping we can license the backbone separately, so then all we have to do is when we pop a new spike gene in there is go through a minimum process of efficacy testing and safety testing. So when we did this, we verified that the rescue virus was indeed our virus because we had changed the codon usage of this particular clone. There's a lot of stumbling blocks with infectious clones. One of them is you have to recreate the pieces of the virus in E. coli. E. coli doesn't like the sequence of this virus very much, and so we have a lot of problems with E. coli wanting to change things on us and kick stuff out. So we did optimize it for E. coli. When you do that, then you cripple it for growing in eggs. So we've gone back and decodon optimized certain parts of this particular infectious clone so it will actually grow better and be maintained in eggs for us. The other one the last one I want to talk to you about is what we call pseudo-typed viruses. So what we do here is we take a virus, vesicular stomatitis virus, and we actually put the bronchitis virus spike protein on that virus. Okay? Now, the VSV virus is crippled, so it will not replicate. Okay? But we can replicate it in the laboratory, and we can insert the spike gene from any bronchitis virus that we have. So again, we have a virus that's circulating in the field. We sequence that particular spike protein from that virus. We synthesize that gene, and then we can put it into this pseudo-typed virus. They're completely safe because they don't replicate, but they don't induce quite the strong immune response that a live replicating virus would produce. So we're looking at maybe some immune modulators to associate with this, or maybe some other live vaccines that could potentially help this one with the immune response. It's really easy to do. It's not very efficient, but it is easy to do. We take the plasmid, we put our gene that we've sequenced and had synthesized into that plasmid. That plasmid, along with the VSV, glycoprotein deficient virus, goes into cell culture. When the plasmid produces the spike protein and the virus replicates in cell culture, the spikes from the bronchitis virus are put onto the surface of that VSV virus, and then we can then use that as a vaccine in the birds. And this just shows you some of the data that we had where we took the Georgia 08 spike sequence, and we expressed that, and we put it in VSV, and that's an electron micrograph picture of the virus that we got back out. Okay. So right now we're in the process of putting this into birds to show its efficacy, and we'll have that data hopefully here fairly soon. Okay, so just to reiterate, we vaccinate to protect against clinical disease, but we also want to stop replication of this virus because we don't want these variants emerging. Every time it replicates, it can generate these mutations. And also, if we can control this disease, we can reduce the secondary bacterial infections that we see in birds, and that can give us a way to use less antibiotics. So homologous vaccines are the most important thing. That's what we want to use to protect. That gives us sterile protection. It stops the replication and the transmission. Licensed vaccines many times don't protect against the variant viruses that we have out there, so these new methods for controlling, they generally are looking at more quickly attenuating the virus by quickly attenuating it or actually engineering a vaccine for the strain that's circulating in the field. And all this is really sort of the base or foundation needs to be surveillance. We need to know what's circulating out there before we can actually make a vaccine against it, right? So surveillance is really key with this, and we've got some really good diagnostic tests that we use for this particular virus that tells us the type. So that's what I wanted to present as far as the work that we've been doing in my laboratory and the control of this virus. I threw a couple slides in here right at the end because the 2019 N coronavirus, or Wuhan coronavirus, this novel coronavirus, has been in the news all over the place. This data was taken in January 26th. The virus was first detected in December in Wuhan, China. It initially spread to other individuals. That seems that it came from animals in one of the wet markets that are there in that particular city. The symptoms are cough, fever, shortness of breath. It's got anywhere from a 2 to 4 day incubation period, and you can see the list there of the different countries that have been affected. That's already outdated. I was hearing on the news this morning that there's over 7,000 people now that have confirmed infected and 170 deaths. So the full genome is available for this particular virus. There is an RT diagnostic test, a real-time PCR diagnostic test for it. In the US, as far as I know, there's still only 5 confirmed cases, but there's quite a few people that are under surveillance at this time. And again, the number of cases there is, I did that yesterday at 1 o'clock in the afternoon, and it's already over 7,000. So those were the numbers as of yesterday at 1 o'clock. Let's put some perspective to this, though, okay, because there's a lot of people that are worried about this. Seasonal flu, the fatality rate for seasonal flu is 0.01%. The annual deaths for flu vary anywhere from 290,000 to 650,000 worldwide. Okay? SARS, you guys remember SARS a few years ago? Okay, SARS had a fatality rate of about 9.6%. The total number of deaths were 774. MERS was the other coronavirus that came out of the Middle East. The mortality rate for that was much higher. It was 34.4%. Total deaths were 858. The new Wuhan coronavirus, the 2019 N coronavirus, the mortality rate for that is 3% so far, so fairly low, but it is much more infectious. It's very, very infectious. And that's the scary part about this particular vaccine. The infectious rate for this vaccine is somewhere around 2 to 3.4 or 5%. Okay. What that means is one individual can infect 2 or 3 other individuals. Okay. Let's put that in perspective. The R0, or that infectious rate for infectious bronchitis virus, the virus we work with, has been calculated to be around 19. So you think about, you got 50,000 broilers in one house, the R value is 19. It's very infectious. Okay. And my question, what about a vaccine? We don't have a vaccine for SARS. They haven't developed that yet. Antivirals, we don't have any of those for SARS either. So we'll see. Right now it's test and quarantine. That's it. Thanks, guys. Appreciate it.